All right, our next speaker is Dr. Rick Diz from Gannon University, and he's going to be presenting on nutrient loadings from the watershed.
And as it happens, I've got a different version of this file.
Oh, I'm sorry.
No, it's not your fault. I just loaded it earlier when I came in. There we go. I feel very constrained by the microphone and is it battery?
Yeah.
Oh, excellent. Because I just hate standing behind that thing. So you heard my name mentioned several times already, and one of the real joys of being a research scientist in Erie, Pennsylvania, is getting to work with Mike Gannon. I think we started working on stream projects 15, 18 years ago, and I look forward to every session where we got together to go out on that boat, and every other time that I've had the opportunity to work with Mike. I don't think I've ever known anyone who took such joy in collecting samples. And I just find it so invigorating and inspiring. So, but I only have limited time. Who holds up the...
That would be me.
Oh, okay.
I'll give you a 2-minute warning.
I appreciate that. Okay, thanks. As Mike explained, he and I are partnering on a project that's being funded by the Sea Grant program, and of course we're very grateful for that opportunity. It's a long project, and there are several component parts to it. Dr. Campbell is the biologist and is responsible for and in charge of analyzing those samples that we collect out there for the organisms, and now I understand that he's foisted that off on Angelia, that it's really her job. Maybe I'll get some of those numbers sooner than I did last year as a result of that, Mike. So you already have some numbers. That's good. I'm the water quality guy, the hydrology and water quality guy. So we complement one another's areas of interest and expertise quite nicely. And I'm limiting my talk really today to nutrient inputs from the watershed. And I have only a little bit of information about what we did in the bay. We're still a long way from having any real conclusions. This is one of those images that Mike referred to. This is probably the most recent image that NOAA has posted on their website about HABs. It's really interesting, isn't it, to see this bloom of, we assume microcystis, that they sort of claim that that's microcystis. I find it quite interesting that the flow coming down the Detroit River here is sort of pushing it away, and it's almost like a flame coming out of a Bunsen burner kind of thing. Just the sheer force of the water apparently is pushing that algal bloom away from Detroit, which of course is right there, and is apparently fed by the Maumee River, which comes into the lake right there and deposits large amounts of nutrients that mix with the very nice high-quality water coming down from the upper Great Lakes, and then in this sort of soup pot kind of environment leads to these blooms. This is what is this little bay right here? Sandusky Bay, yes. And in passing, they always mention, well, Sandusky Bay has a bloom. They don't seem to be very interested. Unfortunately, these satellite images don't tell us much about our, but this is another graph that's presented on that NOAA that now has a bar column for this summer season, indicating the relative severity of this year's events combined, consolidated compared to prior years. We see this disturbing trend in the western end of Lake Erie, where it appears that things are getting worse. And of course, none of us are happy about that. And we certainly don't want blooms of these organisms in Presque Bay either. So we are similarly concerned. The question about temporal variation and Dr. Campbell's observation about how much difference it makes from one day to the next. This is from, as you can see, from the summer of 16. This is chlorophyll concentration derived from the DEP's buoy that is moored in Presque Bay from about early May through the fall, through September sometime. And as you can see, the remarkable and dramatic changes in chlorophyll concentrations from sometimes dramatic and extreme changes just from one day to the next. It's really quite remarkable. I never expected to see that kind of behavior. And so we had, this red is the water temperature of Presque Bay. That buoy also records water temperature, several other parameters it also measures. And it's been very valuable to me in trying to provide a backdrop for what we're doing. So here we have a bloom of something that's green, another little bloom, a bloom here, it goes back down, goes back up, and then look at that. That's just incredible. That's essentially all on just a single day. Numerous measurements. Of course, this records a data point every 6 or 10 minutes or 20 minutes or something like that. 20 minutes. Yeah. Now, this doesn't tell us what's causing that, but it's something green. That's all we really know so far. So the goal of our project is to try to understand the factors that would lead to a bloom of harmful algae. That sticks in my throat a little bit. Cyanobacteria are not truly algae, but we tend to call them that. They're photosynthetic bacteria, very primitive organisms. We have several parts of our project. I'm going to talk mostly about my study of water and what it contains coming into Presque Bay from the watershed. We care about nutrients a whole lot in trying to understand this aquatic ecosystem because we assume that there are only certain factors that are limiting. And nutrients, and when we use that term, we mean nitrogen and phosphorus primarily because they are scarce and often one or the other limiting to the growth of photosynthetic organisms that get their energy from the sun. And the response of cyanobacteria compared to the true algae is complicated and not entirely understood just yet. And it might be a number of ways of looking at nutrients that control the appearance of these sudden blooms of organisms and then their sudden disappearance. It might be an interaction of several factors, and that's why we're hoping to use an ecosystem, an aquatic ecosystem computer model sponsored by the US EPA that hopefully will help us organize and synthesize the information we've got. Temperature is certainly important. Folks like to dispute the issue about climate change. I don't know what's going to happen 10 years or 100 years from now relative to the water temperature of Presque Bay, but that might be a very important factor since cyanobacteria have an optimum growth rate at warmer temperatures. And the other bacterial group, algal groups, typically, generally, prefer cooler water temperatures. This is that same chlorophyll graph, but in this case I've overlaid a few of the data points that Dr. Campbell provided to me for certain of our trips that we made out into Presque Bay last summer. This is in milligrams per liter, biomass in milligrams per liter, not counts. Cell counts and biomass concentration are different because some of those cells are very small, some are big, so just the number alone doesn't tell you everything that we might need to know in terms of nutrient uptake and growth rates and things like that. The purple dots here are cyanobacteria. These brown squares are diatoms. Traditional thinking says that diatoms don't like warm water temperatures, but here we are at the end of July, early August, and we've got what appears, apparently this bloom right here, perhaps a combination of cyanobacteria and diatoms. We would expect that this out here would be diatoms because diatoms seem to do very well in cool temperatures. One other interesting thing here is that the so-called green algae, which I thought would have been more abundant, very low levels, at least in this data set that I presented there. So I'm going to describe what we did in the few minutes I've got left to monitor the streams coming into Presque Bay from its watershed. This table shows the more important streams and their length and the area that they contribute as a percentage of the total watershed. I had monitoring equipment on these two streams, Cascade Creek and Mill Creek, which together constitute about 75% of the watershed. This is a little map that gives you, for those of you who aren't familiar, gives you an indication of the Mill Creek watershed. That's, remember, about 50% of the Presque Bay watershed. And here we have the Cascade Creek watershed. Most of this is actually in storm sewers. Very little of this is in open streams, except in the very lower limits here, and then in spotty little bits and pieces here and there. This is the sort of monitoring equipment that we used. This is a box located alongside Cascade Creek. As you can see, it was taken last winter. This is inside the box, a big 12-volt battery, and this ISCO sampler. This is all gone now. The storm on Sunday with the tornado and the huge rainfall, almost 3 inches of rain in about an hour or so, completely washed this box away. It has disappeared. The box is gone. The boulders, you remember the boulders? We sort of lodged it, gone. All those boulders are gone. I could not find the equipment. I went downstream searching for thousands of dollars worth of scientific equipment, and totally gone. So in order to quantify the loading of chemicals or substances into the bay, we have to know how much water there is, and then we have to know what's in the water. So the monitoring equipment simply measures depth. You then have to convert depth to flow rate or discharge, which is volume per unit of time. And that all by itself was a huge challenge because this data set is complicated. But these are the stage discharge relationships that I developed for each of the two streams. These are based on something called Manning's equation. If any of you know any hydraulics or hydrology, these are based on Manning's equation, then sort of factors that are consolidated and simplified so that I could use them a little bit better. One of the complications is that the monitoring site for Mill Creek, because it's in a tube, for those of you who know the area, Mill Creek is in this huge pipe until it emerges at the Bayfront Highway down at the wastewater treatment plant. So if we wanted to get the flow through the city, we had to be below the mouth of the tube, which meant we had to be down at the wastewater treatment plant, which is very close to Presque Bay. Well, Presque Bay goes up and down all the time. Presque Bay and Lake Erie go up and down all the time. It's sort of like a tide, but it's driven by winds. It's called Seiche behavior. And this up and down, up and down, up and down. So this is an overlay of Presque Bay or Lake Erie's level fluctuations along with the same data set from my monitoring station for that same time period. So figuring out what the flow rate in the stream is when the water's going up due to Lake Erie going up, and then down when Lake Erie goes down, was quite a challenge. So I had to figure out a way to subtract out the influence of Lake Erie on the monitoring station. This is a storm event. You can see these peaks where the blue goes up quite a bit more than the green, sort of anomalously it goes up. That's a storm event, and that's a storm event. But here we had dry weather, so the monitoring station just followed the lake level, essentially. Rainfall is the driver of all this, and it's quite variable. And so this blue line shows the average normal rainfall in Erie during, if there ever is a normal summer. The sort of rust-colored bars are the summer of 2016, month by month. You can see it started out dry and then it got wet at the end of the summer. And then this past summer, we had a very wet June, including another huge storm event at the end of, well, the middle of June. Then it got quite dry. So the hydrology of the streams is driven by the rain, which was quite different for the 2 summers that we have been studying. This is a summary. This is just for the summer season, June to September. These are the results of my calculations for how much water left each of these streams. I made an assumption, 2 minutes. I made an assumption that the Cascade Creek watershed was representative of the unmonitored part of the Presque watershed, and just prorated that quantity to represent the unmonitored portion. So when we had almost 19 inches of rain during that summer, my calculations show that we had 13.7 million cubic meters of water leave the watershed and go into the bay. But this past summer, with less than 15 inches, it was about 11.5. The water per square mile is interesting. We're getting more water per square mile out of the Cascade Creek watershed than we are out of the Mill Creek watershed. That sort of makes sense because the Cascade watershed is much more urbanized, and the Mill Creek watershed has a higher percentage of what we call pervious surface, where water can infiltrate into the soil. I pointed those numbers out there. This is just a history of storm events and the discharge in Mill Creek and Cascade. That red line is the rainfall. You can see the rainfall and the flow in the stream correspond quite tightly with one another. This is for this past summer, where we had these very sharp increases in flow in the stream, and then it goes right back down again. These are very urbanized streams that have very flashy behavior. Now, we also measured nutrients and other substances in that water. The ISCO samplers were programmed to activate when the water level rose. So for the number of storms that we were able to collect samples and analyze them, in 2016, we only had 4 storm events that we were able. This is in addition to measuring water from the bay, where we went out every 2 weeks and we're analyzing bay water. We were also trying to analyze stream water. So we had 4 events for Mill Creek and 4 for Cascade. They for some reason, they weren't exactly the same ones. Phosphorus is the thing that we mostly focus on. This is TP is total phosphorus. Total phosphorus is more than just phosphate. It's all of the forms of phosphorus in that water sample. The dissolved total phosphorus number is after that water sample was filtered. So the particles are removed. And so you can see 0.34 milligrams per liter in Mill Creek and 0.3 in Cascade Creek water. Nitrogen may also be important. We don't know that yet. I tried to focus more on the streams this past summer, and so we were able to get more storm events. And now remember, the rainfall was different. So there was less rain, so maybe the chemicals were more concentrated, 0.37, but this is a strange number. This jumped all the way up in Cascade Creek to 0.56. I double-checked that several times. This is just a summary to more readily compare those. I can't quite stop. I have to keep going. So to better understand where the phosphorus is, this shows the blue is the dissolved phosphorus and the brown is the particulate phosphorus. So you can see that in both streams, about 3/4 of the phosphorus is attached to particles. This is an individual. This was all the storm events. This is an individual storm event with samples collected every 10 minutes during that storm event. You can see that the dissolved concentration stayed pretty much the same throughout the entire event. But the particulate concentration went up and came back down again as the water became very, very turbid, and then as the storm event was ending, became less turbid. We then used concentration and flow together to calculate mass. And so these indicate the mass loadings of these chemicals to the bay. And notice they're very much storm dependent. So if someone can figure out how to trap the particulate matter during storm events, Amber was going to figure that out, that would be great. Because it's mostly during storm events. That's a huge undertaking. This is another way of looking at those numbers, and I show here the production rate per square mile of phosphorus. Notice that Mill Creek produces a little bit more per square mile in 2016, but less per mile in 2017. Remember, the amount of rain was different. This also has to do with that very high concentration during 2017. Nitrogen, less rain, higher production per square mile. Very strange. I don't understand why. This is a schematic diagram of the aquatic ecosystem computer model that we're going to use to try to assimilate all of this information. And we still have lots to do. Dr. Campbell and Angelia are working on the plankton. I'm going to be working on the aquatox model, and then once we get it to work, whatever that means, then we'll be able to speculate by doing so-called what-if scenarios. Sorry that I couldn't stop.
While we're loading up the next talk,